Welcome back. We are looking at a transfer coefficient approach. We had taken the specific example of oxygen transfer in bioreactors, oxygen supply to bioreactors. After a lot of discussion, we have come up with this formulation. The moles transferred per unit volume per time is a certain Kxa times Xa star minus Xal. Xa star is the liquid side concentration of oxygen, which is in equilibrium with the bulk gas side concentration. And A is the interfacial area per unit volume. We said A is of course difficult to measure, and therefore Kxa is measured together. Kxa is somewhat equivalent to Kla, only thing is that Kxa is written for mole fractions. Kla is usually used with concentrations. The total concentration times the mole fraction is the concentration of the species, and therefore we use and we use Ka quite extensively when we deal with bioreactors. So what I'm going to do in this class is to tell you how to estimate Ka because it is important, and also we'll work out a problem that would help you understand how to estimate Ka. Okay. Let us see how we're going to go about it. Some methods exist to estimate KLa in bioreactors, some standard methods. And the KLa needs to be estimated at the conditions of the cultivation or fermentation. Why? Because it depends on temperature, it depends on operation condition, and so on and so forth. So you fix everything. You fix the temperature at which you're going to operate, you fix the pH at which you're going to operate, maybe also. And you fix the RPM and other things that you're going to operate at, and then estimate the KLa. There are two broad methods: sulfide oxidation method, dynamic response method. What I'm going to call as dynamic response method here is a certain method. This name has been used in different ways in different textbooks. Okay? So please be a little careful when you come across this term. Dynamic response method might refer to a slightly different method in some other textbooks. So let me call it dynamic response method. Sulfide oxidation method can be used, but there are some limitations of this method. I just thought I would mention the name to you, but I'm not going to tell you anything about this method. Okay. So let us look at the dynamic response method as it is called in this course. The basis for the dynamic response method is nothing but the mass balance on oxygen over the system. Okay, it's also being used here, and that's the reason why we started looking at mass balances as some sort of a review to begin with. Okay. The system over which we are going to do the balances is the bioreactor broth, and we are mentally going to remove the bubbles from our broth. Okay. So broth minus the bubbles is going to be our system. That we can focus on. It can be a conceptual system, right? So this is going to be our system for analysis. This comes in handy. This we'll know only by experience. I will not. It's a little unrealistic to expect a novice to come up with this choice of a system right in the beginning. This comes out of experience after you've been hit upon by various different difficult choices of systems for the purpose at hand, and then you come up with this elegant system or this way of choosing a system by which the analysis becomes elegant. Okay, so take it on face value. We are going to do it over broth minus bubbles. We have taken the bioreactor broth. We have mentally removed the bubbles from it. The mass balance equation we all know by heart now. Input rate minus output rate plus generation rate minus consumption rate equals the accumulation rate, and we are writing this on oxygen. This is a bioreactor system, broth minus bubbles. The output rate is 0 of oxygen. Now recall, this is the bioreactor broth. The oxygen is being bubbled, or the air is being bubbled through the bioreactor broth, through the bubbles, and we are looking only at the liquid part of the broth. So this is our system. Oxygen does not get out of the liquid part of our system until supersaturation conditions are reached. right? That's the only way by which it can go out of the liquid. And we normally don't operate under those conditions. That's the reason why this can be put to zero. And this is essentially the reason why we chose this as a system. We mentally removed the bubbles so that we can put our output to zero. If we had included the bubbles, there was a lot of oxygen that was going with the bubbles. Okay, we needed to account for that, and that would have become a lot more difficult to do, a lot more measurements to make. So we excluded the bubbles, and we could put RO equals 0. The generation rate, of course, there is no reaction that is generating oxygen in the system, and of course, input rate is there, consumption rate is there, and accumulation rate could be there. What is the input rate? Now we have an expression for the input rate in terms of KLA. KLA into CO star minus CO, CO2 star minus CO2. Now note, KLA, the volumetric mass transfer coefficient, times the concentration difference here. Okay. This is in terms of moles transferred per unit volume per time. We want each of these in terms of moles transferred per time, mass per time, moles per time. We can work with moles per time, and there will not be a big difficulty in this case. There is no reaction anyway. So we want moles per time for each of these remaining terms here. This is KLA CO star minus CO, and because this is in moles per volume per time, we need to multiply it by the relevant volume, which is the broth volume. So now we have it in terms of moles per time. Okay. Why? Because the input is across the interface between the gas bubbles and the liquid. So that is the input to the system. Okay. Now the consumption rate, we are going to write in terms of a certain oxygen uptake rate, QO2. The units of QO2 are such that you need to multiply it by the cell concentration, okay, because it is normalized with respect to cell concentration, and also the volume to get it in terms of moles per time of oxygen. Therefore, QO2, oxygen uptake rate, times the cell concentration times the volume, equals, of course, this is mass of oxygen. Mass of oxygen is concentration times volume in the system that we're looking at. Therefore, ddt of mass of oxygen, which is CO2 times V. The volume happens to be a constant, and therefore that can be taken out of the derivative. Also, let us take the case when no cells are present in the bioreactor product, x equals 0. So when no cells are present, x equals 0, and at constant volume, V is a constant, we have KLA CO star minus CO2 times V. This term drops out, x equals 0. Now V is a constant, it can be taken out of the derivative, V times ddt of CO2. So this V can be cancelled and written KLA times CO2 minus CO2 star minus CO2 equals ddt of CO2. If you solve this equation, right, this is a differential equation, right? If you solve this equation with an initial condition, of course, you get dCO2. This is a long term here. Recall dCO2. You need to take concentrations to one side. So you have 1 by CO2 star minus CO2, dCO2. So the long term arises because of this. And this is straightforward KLA dt. So a T term comes here. So dCO2, CO2 star minus CO2 equals KLA dt. And from the table of integrals, we need the integral for the form. This is equivalent to dx by a minus x in the table. Okay. Dx by a minus x. This, of course, is a constant, as you can recall. Therefore, the solution of this form you can directly take from the table of integrals. You don't have to worry about working it out yourself. Ln of C star divided by C star minus CO2 equals KLA. Okay. So this is the solution from the table's standard form. And what does this mean? So this is of the form y equals mx, okay, where y is nothing but ln of C star divided by C star minus CO2. So if you plot ln of C star by C star minus CO2 versus time, you will get KLA as the slope. Okay. So this is the way of getting KLA in an actual experiment in the industry and so on and so forth. The way it is done is as follows. We plot, we look at, we measure, that is, we measure and then plot dissolved oxygen in the broth versus time. And the experiment we do is as follows. We first purge the liquid with nitrogen to physically strip out all the oxygen that is present in the broth. Okay. So you pump it with nitrogen, that nitrogen replaces the oxygen in the broth, therefore the oxygen concentration in the broth drops gradually till it reaches zero. Make sure it is indeed zero, wait for some time, and then at time t equals 0, introduce air. Air is being introduced here, and then because of the oxygen coming into the broth, the DO level is going to increase. It will increase something like this and reach a saturated value. Okay. So once you have this data, you could plot this quantity ln of C star divided by C star minus CO2, because you have CO2 versus time data, right? So you can plot the entire data and you have that power of the entire set of data to get a good estimate of KLa. It will be the slope of the curve ln of C star by C star minus CO2 versus time. Let me present this problem to you and leave it to you to work out. When we come back, I will present the solution, okay, in the next class. Yeah, I think that might be a little better. The following data was obtained during KLa determination of a stirred tank bioreactor operating at 500 rpm, 1 atmosphere pressure and 37 degrees C, and the measurement is done by the dynamic response method. Okay. Note I mentioned about the conditions of estimation being the same as that of operation. So you need to fix these 500 rpm, 1 atmosphere pressure, 37 degrees C, and measure KLa under these conditions. The oxygen source was air. A millivolt meter was used to read the dissolved oxygen level, and this is typically done in labs, in the industry also, because the voltage is proportional to the concentration. The DO probe converts the concentration to a certain voltage, and the voltage could be either directly measured or you could use some sort of an interface device to convert it into the concentration. We had used, this is actually data from the lab, and we had used a millivolt meter to read the dissolved oxygen level. Find the KLa of the bioreactor. This is the data actually that I had generated during my PhD days. Okay, this is actually data from my PhD work. I used to run tons of bioreactors, a large number of bioreactors for my work, and this is during characterization of one of those. Okay. So the data that is given is the time in seconds versus dissolved oxygen in millivolts. So this is proportional to the concentration in either molar or in terms of percentage air saturation, whatever you want. Okay. So you can take this problem. I've already given you the procedure for working this out, right? You plot ln of C star divided by C star minus CO2 versus time and get KLa as its. Why don't you try this out on your own? It's good because it's useful, comes in handy for many different practical situations. Why don't you try this out? When we come back, and that's what I have here. Yeah, when we come back, I will give you the solution, or I'll start out with the solution for this. See. Bye. Welcome back. In the previous class, we looked at a method of measuring KLa, the dynamic response method, and I had posed this problem to you. I had asked you to solve this as homework, and let us start by solving this one. The following data was obtained during KLA determination of a stirred tank bioreactor operating at 500 rpm, 1 atmosphere pressure, excuse me, and 37 degrees C by the dynamic response method. The oxygen source was air. A millivolt meter was used to read the dissolved oxygen level. Find the KLA of the bioreactor. You have the data here, T in seconds was DO in millivolts versus T in seconds. Okay, we already looked at problem solving at the very beginning of this course. So we look at what is needed first. In this case, KLA of the bioreactor by the dynamic response method. What is known or given? We have data on millivolt versus time during the oxygen sorption process in the bioreactor in the absence of cells. Millivolt can be directly related to oxygen concentration in the broth or DO. If you want, you look at the DO probe working principle from your bioreactor course, or you can read up about the DO probe working principle. You will get why we can use millivolt in the place of actual concentrations. So what is needed, what is known, we are clear about. Now how do we connect these two? And of course also known as the conditions of operation, that is fine. And how to connect what is needed to what is given. How do you connect? We have already derived that ln of C star divided by C star minus CO versus time will give KLA as a slope. So this is the connection between the two. Okay. So let us see how to do that. I've already done that in a spreadsheet, and so let me go to that directly. You see here, can I increase the view size? There are views here, yes. View zone. Let's go to maximum zone. Okay, here we are. Okay, all these things are nice to do with a spreadsheet. This is the data that is given. In the first column, you have time in seconds. In the second column, you have millivolts. Okay. I've just considered the relevant times here again for ease of plotting, same column here. And here you have ln of C star divided by C star minus C. You need to find the C star value. Right? What is the C star value? It is going to be 1.1. Why? Go back to this. Here, what is C star? This is the value that is in equilibrium with the gas phase concentration, right? So you see here the data, in millivolts itself, 1, 1.1, 1.1, 1.1. It has reached saturation here, okay, or it has reached equilibrium conditions here. So this is the value that you're looking for. The value in the gas phase that is in equilibrium with the bulk phase concentration in the liquid phase, right? So this is the value that you're looking for. So you have a measure of that itself, so you can directly use it. If you don't have a measure of that, you'll have to measure the saturated value and use that for C star. That's as simple as that. So let's get back to this Excel. So I've calculated ln of C star 1.1 divided by C star minus C. The C is given in as various DO values here in millivolts. I'm using all millivolts, therefore the units are consistent here, and the variation is given here. And of course, this doesn't make any sense, therefore don't worry about that. So you can use this set of data, ln of C star divided by C star minus C versus time, to get KLA as a slope. So if you plot this, it is going to look something like this. Okay. Now you take the linear region. You have 1.1s coming in here, right? So you need to avoid that. And the linear region is somewhere here. You take the linear region because the relationship is valid only in the linear region. The process itself goes over various regions of description. Different parts can be described by different ways. Only one major part can be described by linear variation. And if you do that, the actual plot is something like this. Let's again zoom in. Can we? Yeah, this is what it is. You have ln of C star divided by C star minus C versus time here, right? And the plot is somewhere here. We have neglected the other points where this relationship, the linear relationship, is not valid. Therefore, we need to look at the data points only where the linear relationship is valid. Okay, this is not fudging data. This is applying the model to the region where it is applicable. Okay, we are not arbitrarily removing any points. We are removing the points which does not fit or which are not described by the model. So you have this here. The equation turns out to be the fit 0.0914x minus 4.9609. The fit is reasonable. R squared value is 0.943. And you take the slope here, it is going to be 0.0914. This is in second inverse. And in R inverse, we need to multiply it by 3600, 329 hour inverse. Okay. So 329 hour inverse is actually a very high K. Of course, we had used a diffuser, a HPLC diffuser for diffusing out our bubbles. We used to have very fine bubbles that way. So the surface area per unit volume was very large. And our RPM itself was 450 or something like that. That is a yeast bioreactor. And therefore, this high KLa is acceptable. Okay. Fine. Now, should we do this here? I think why don't we take it up in the, I know this is a short class, but why don't we take it up in the next class? Because we're going to discuss some papers from our research. I'm going to tell you a lot of places where I've actually applied the transport principles in research, in cutting edge research. I thought it would be good to give you some examples. We have worked a lot on these aspects. See you.